You probably noticed that I did not put another set of slides up on Canvas this morning, and that's because I realized that with getting through the next two framing questions, and I want to work out some problems with genetic structuring that we weren't going to have time to get any further before our exam on Friday. So this material is not on the exam. Hopefully that was made clear by the study guide. But in case it wasn't, this material is not on the exam. It will be on the next exam, which is the Friday right before finals week. And so I think that might be it as far as announcements go. Yeah, Jake.
Is there anything from Monday that will be on there?
No. Well, yeah, because on Monday we actually finished up the chapter 29. And so the exam covers chapter 28 and 29. So we did finish up chapter 29 on Monday, and that portion will be on the exam. Okay. I think that's it now. Unless there are any questions on structural stuff. Yeah, Anna.
Is that in 29 when we talked about the lecture break with the
Yes. Yep, absolutely. Okie dokie. Well, let me pray for us, and then we'll get started. Father, thank you so much for just how you encourage us, and I know that I need it right now, and I'm sure everyone in this room needs it. Father, I pray that as that encouragement comes, that we would use the hope and the joy and the energy that comes along with that to serve you well and to push hard for the remainder of this semester. Father, remind us that every opportunity we have this semester is an opportunity to bring you glory and an opportunity to become more like Christ. Pray that that would be true. We ask this in the name of Jesus. Amen. All right. So last time we finished off talking about genetic structuring, and I want to remind you that genetic structuring it's basically the frequencies of all of the different genotypes that are possible from your gene pool. Now we try to keep our examples simple with just two alleles, two forms of that gene, one dominant and one recessive, because it keeps things really nice mathematically, makes it to where you don't have to do some higher level calculations. So if you're ever given an example beyond that, I would give you some more information and not require you to actually work through that. But so whenever you're talking about genetic structuring, you have something that is actually a really nice baseline by which then you can compare how does this population change from generation to generation to generation. Is the genetic structuring remaining the same? That is, does each genotype represented in your genetic structures, your genetic structuring, are those proportions remaining the same generation after generation after generation? Because if they are not, that population is changing. It is microevolving, right? It is, there are some evolutionary changes. So we're going to talk about a mathematical formula called the Hardy-Weinberg Equilibrium that basically demonstrates how you can maintain genetic structuring. So yeah, this model demonstrates when genetic structuring is going to remain the same generation after generation. In addition to that, it gives you the ability to understand even if your genetic structuring is remaining the same, but it doesn't match up exactly with your allele frequencies, it gives you some indication that something strange is going on. Something strange is going on. All right, let's talk about some of the assumptions of the Hardy-Weinberg Equilibrium. Okay, one, it assumes that the population is infinitely large. And this is an interesting assumption because obviously a population cannot be infinitely large. Okay? But it brings up something that's important. That one, obviously this assumption cannot be met. However, it demonstrates that there's something about population size that is important with regards to evolutionary change. Okay? So keep that in mind. Store that up somewhere, that the size of the population has a big impact on the potential for the population to evolve. Okay, another thing it assumes that there are no individuals leaving or entering the population. And this is an important one because even if the individual is only bringing a genotype that's already there, right? So the individual's not bringing any new information. It's not bringing any unique allele. Or if the individual is leaving, it's not like you're only representative of that allele in the population leaving because it's not wanted, right? It's even if that is not the case and you're only bringing in what's already there, or you're only losing what's there in pretty good numbers, it still is going to change your frequencies, right? Even if you have 1,000 individuals and 750 of them have the exact same genotype, if one of them leaves, your proportion is different now, right? It's no longer 750 out of 1,000. Now it's 749 out of 1,000. Okay, does that make sense? Okay, so that may not sound like a big deal to you, but it's a big deal. It's a big deal. We'll talk about why it might be a big deal under certain conditions. It also assumes that there are no mutations. No mutations introducing unique alleles. And oftentimes what you find is the recessive allele is the product of some kind of a mutation. You find that to be the case a lot, especially with regards to genetic illnesses, where the dominant allele, let's use color blindness as an example. Color blindness, great example. We'll talk a lot more about this next semester in cell biology. But color blindness, red-green color blindness, is basically the result of a single gene. Okay? And so the normal allele allows you to differentiate red and green. Kind of important, right? Not life-threatening if you are red-green colorblind, but it's kind of important. The mutated form of that gene makes it impossible for you to differentiate red and green. And so a very, relatively common mutation just converts one of those alleles into the other. So this assumes that there's no introduction of new alleles through mutations. It also assumes that you're not changing the allele frequencies because of mutations. Maybe a common mutation that converts the dominant into the recessive. Also assumes mating is random. And what this means is that there's no individual that is more likely to produce offspring than any other. That every individual basically has the same probability of finding a mate and producing offspring. Another way you can phrase this is that there is no sexual selection. So there's not some feature in in the sexes that makes them more or less likely to produce offspring. And then lastly, no phenotype is favored over any other, and that is that no phenotype, no physical appearance, right? No physical form survives better than any other, right? The environment does not favor them in any way. They're not outperforming others.
So no natural selection there.
No natural selection. Yep, that would be another way of saying this is no natural selection. All right, how likely is it that you are going to meet these assumptions? It's impossible, right? You cannot even meet the first assumption. It is literally impossible to have an infinitely large population. What does that even mean? I don't know. Infinitely large. However, what you find is that populations often are at Hardy-Weinberg equilibrium that demonstrate that basically all the forces at work are balanced and that the population is not changing in any significant way, which is kind of interesting. Even though it's it is impossible to meet the assumptions of the Hardy-Weinberg equilibrium, you find that most populations actually are at Hardy-Weinberg equilibrium. However, assuming they are not, so because populations are always being influenced, if they aren't balanced, this equilibrium basically gives us a baseline that we can compare an evolving population to and try to figure out which of these assumptions is not being met. Now, the first one's easy, right? You're like, the first one can't be met. So obviously the first assumption is not being met. However, this again is a reference to the role population size plays in influencing evolutionary change. Yeah, Joey.
So, I mean, is it analogous to like when you're in a science experiment and you have a control and then you have like the one where you're changing variables? So like,
Sure.
Like nothing is there's nothing abnormal happening, and then we can compare it.
Yeah, it's interesting you bring up the idea of a controlled experiment. The problem that you have when you're studying populations is it is almost impossible to do a controlled experiment, right? I mean, if you're going to bring them into the lab and completely manipulate everything, then sure, you can do a controlled experiment, but that's not real life, right? And that's why we have weird things happen in the lab, and you're like, man, look at all these cool things we got E. coli to do and develop, and you're like, yeah, but it doesn't actually happen because E. coli doesn't live under those conditions in real life, right? And so because it's virtually impossible to do a controlled experiment on populations in situ, like where they actually are found, what you do is you try to have this of no change. And this is exactly what it would provide you. That if nothing were acting on this population, this is what it would look like generation after generation after generation if it were not evolving. So it does basically take the place of the control in a controlled experiment. So it then provides you something to compare and to say, okay, well, it's not meeting this control, right, where no change is happening. It is changing. What's causing that change? And the answer to that would depend on how everything is actually shaking out. All right? Does this make a little bit of sense? Is it okay? Okay. All right. So here's a figure from the text, and it basically shows you how you can go from genetic structuring. So here it's giving you the proportion of individuals that are homozygous dominant, that have 2 dominant alleles, proportion of individuals that are heterozygous, have 1 dominant, 1 recessive, proportion of individuals that are homozygous recessive, have 2 recessive alleles, and then to move from that to get to the allele frequencies. And then at the bottom, it shows you the opposite, how you go from the allele frequencies to figuring out what your genotype frequencies should be. And this is representing what's called a Punnett Square. We haven't talked about this, and we won't talk about it a lot until next semester, but whether you've experienced it before or not, basically what it's demonstrating is a cross between 2 individuals. Because both these individuals are diploid, they basically can make gametes with potentially 2 different alleles if, of course, they have 2 different alleles, right? We are diploid. Yes? I'm diploid. I assume you are. I've not seen your genetic makeup, but I assume you're diploid. And so, statistically speaking, if I did not know what gene you carried for red-green color blindness, and this one isn't actually the best example because it actually occurs on the X chromosome, which males only have 1 of, and females have 2. So let's just restrict this discussion to female students. You have 2 X chromosomes in each cell. Assumably, you may have more, but you should have 2 in each cell. And if you did not tell me whether or not you are red-green colorblind, and I was just going to assume, I would say that, there's a, whatever the percentage of alleles, total alleles made up of the dominant, I would assume that that's the probability of you producing a gamete with that allele, if I didn't actually know your genotype. And then I would assume whatever the proportion of the gene pool made up of the recessive allele, I would assume that that's the probability that you could produce a gamete with the recessive allele, because I don't know your genotype. I have to assume. And so that's what this is doing. It's crossing 2 individuals. You don't know their genotypes, so you have to base it entirely on probability. Okay? If 70% of the gene pool is made up of the dominant allele, you would say there's a 70% probability that that individual is going to produce a gamete with the dominant allele. 30% of the gene pool is made up of the recessive allele, you'd say there's a 30% probability that they're going to produce a gamete with the recessive allele. Okay? And then we represent this in the crosses, and then we just work through from there and see what genotypes are possible. So here would be a cross that produces an offspring that has 2 dominant alleles, right? And if the probability of the allele being dominant is 70%, the probability of producing this genotype should be 70% times 70%, which would give you 49%, or 0.49. You can do that same thing for the heterozygous and for the homozygous. So this is just illustrating how you go from one to the other. During our lecture break, we're going to do some of these. We're going to put some pencil to paper, going to get some cramps in our hands. It's going to be awesome. It's going to be awesome. You're going to love it. I'm going to love it. Even your children are going to love it when you tell them the story of how great your experience was on this specific day in your life. That was hyperbole. So, anyways. All right, any questions here? So you know genetic structuring is not the allele frequency, right? Genetic structuring is the genotype frequencies. And we'll keep all of our examples to be a simple setup. Not simple meaning it's not difficult at all, but simple compared to something more complex, where you have two alleles that are possible. So there aren't any questions. We're going to take our lecture break, and we're going to work out some problems. Okay, so I'd like for you to orient yourselves into groups of 6/7/8, and you're going to need a discussion leader, and that discussion leader, instead of getting every single person voice heard, your responsibility is make sure every single person is tracking with what your group is doing mathematically. Okay, a little bit more responsibility for the discussion leaders today. All right, so get yourselves into groups of 6/7/8 and elect yourself a discussion leader.
Did you get that, man?
Awesome.
Okay, then.
Russ, you're the discussion leader.
Oh, I'll show you. Don't you worry. It's not on the slides that are on Canvas yet. I wrote them this morning.
Awesome.
All right, are we ready? Are you in groups of 6/7/8? And you have a discussion leader? And discussion leaders, you're aware of your responsibilities to make sure everybody is tracking as well as you can. Right? If somebody's like, oh yeah, I totally get it. I mean, what are you going to do? Gosh, my children do that. They're just like, they nod their head as you're telling them something, and you can totally tell they're not at all listening. But they're like, oh yeah, I got it. And they're like, what did I tell you? All right, here are the questions. There are 3 of them. All right. If the dominant allele makes up 70% of all alleles, what is the genetic structuring in the population? Okay? And again, all of these are assuming you only have 2 alleles. 2 alleles are possible, dominant or recessive. Second question, well, what if the dominant allele increased to 90% of all of the alleles? And then thirdly, if 25% of a population demonstrates the recessive phenotype, keeping in mind I mentioned this last time on Monday, but that was a long time ago, that the recessive phenotype can only be expressed if you don't have even a single dominant allele. Because a single dominant allele should mask the recessive. That's what it means to be dominant versus recessive, right? Kind of makes sense. Anyways. And then, so if 25% of the population demonstrates the recessive phenotype, what is the genetic structuring? Okay? So number 3 actually requires you to work in a different direction than the first 2. So this should be fun. I'm going to walk around. I'm going to make sure discussion leaders, like there's somebody that's kind of walking through this and tracking, and then I'll work these out. All right?
Okay, so if the dominant allele makes it 75%.
Nope. That's where population genetics comes in handy.
Okay,
For population genetics now comes in handy.
You can do either. Yeah, you don't have to use a Punnett Square. I just wanted to, because we haven't talked a lot about Punnett Squares. I just wanted to explain that so that when you look at that figure, you know what's going on. But yeah, you absolutely do not need to use a Punnett Square.
Okay, so if we
You
have
can do it all without a Punnett
so we
Square.
have A as the dominant and A as the recessive. If you have a Punnett Square, we'll have A. So if you have A here, we'll have
Dominant allele is just one of the 2 alleles that are possible. Dominant and recessive. So if the dominant makes up 70%, and recessive makes up 30%. So there are only 2 choices, dominant and recessive. So you can have dominant be the P, that's what we usually do. Recessive be the Q, and then you go through and figure out your genetic structure. So what would P squared be? What would 2 times P times, yes. What would, there you go. There you go. Yes. Yes. Light bulb.
Yep. The P2 plus 2PQ plus Q2 equals 1. Awesome.
So,
I left my calculator. Oh, the Genotype.
so he just.
Oh wait, why did I
So he
So we gotta go from allele frequency to.
Does everyone know what's going on?
yes, we're using the P2 plus 2P2 and we're just putting in the values.
Let's explain this to Grace.
Cool. So we have
like
70 squared plus 70 squared plus 2, 70 plus 2
times 70.
plus
70 squared times 2.
Yeah, and you just multiply the percentage.
Okay, so the third, so the cube was left over.
So, but
so 30.
You guys have those?
Yeah.
He's in mathematical ecology, so
Yeah.
yeah, he's,
Wait, you did a times? That's a plus right there, isn't it? That one? That is? I got it. Oh, maybe I wrote it down wrong.
it's right here.
No, it is a plus.
Yeah.
Yeah, there's a pay.
So 0.49% is a
Alright, if we're gonna.
So you did it, yes.
Or you can still keep working.
Gonna do 70.
Sorry, Jack. Jack.
No, that's fine. 70 squared. Okay. 49,000. So we have
I'm working it down.
4,900
Wait, that's in
so we have
one?
So that should be the genetic structure.
70 times 30%.
Okay.
Huh?
So do you know how the Punnett Square works? So right, you so 2 dominants, dominant recessive, the dominant.
Because this is. Oh, yeah. I just. I do that and then convert it back. Yeah.
So here you have, I did this, answer right here.
Probability.
What happens with this one? What is it? Should it stay the one, right?
No, it should. It should.
What does this mean?
1 minute.
That's
1 minute.
Oh, so that's percentage.
yeah.
So 49%,
Yeah.
42%,
49%.
and then 90%. Or 9, that's a,
0.9.
that's almost 100%.
That's what you should get. You should get 100%. If you're only dealing with 2 alleles, you should get 100%. If those 2 alleles are in the population. So. Yeah.
Oh, this is,
Yeah.
this
So.
is 9%, not 19.
Yeah. So
Probability, percentage probability.
Ah. Oh, I get it now.
And again, like,
That makes sense. I get it.
30 seconds.
This is a pretty simplified version.
this isn't mathematical ecology.
No?
No, this is population genetics. Yeah. Check that. I had to do a Punnett Square where we had 4 variables.
Oh wow. So that looks like
That's like
and then you get
16 species. Yeah.
That makes sense?
That was the biggest one we got to. Dr. England was like, yeah, I could make you go higher, but I'm not going to.
25% of the population.
Alrighty. Alright. Alright. So, bring it in. If you didn't get a chance to do all of them, it's okay. Because we're going to do it together. We're going to walk through it. And y'all are going to love it. And again, it's going to be one of your fondest memories. Not just of your experience in this class, but of your entire life to this point. Okay, So for this first problem, the percentage of the dominant, so this is the proportion of total alleles made up by the dominant allele, we'll represent this as P, and in this first problem is 0.7, or 70%, however you want to represent it. Doing 0.7 is nice, because it allows us to use this equation. That tells us in a simple setup, where you have two alleles, one dominant and one recessive, the percentages of each need to add up to be 1. Right? So that would make this 1 minus P, right? If P plus Q is equal to 1, Q is 1 minus P, which would equal 1 minus 0.7, which would equal 0.3. So if 70% of the alleles are the dominant allele, 30% of them are the recessive allele. Okay? But that's not the genetic structuring. That's just allele frequencies. This is the genetic structuring. These are our genotypes. So our homozygous dominant individuals are P squared. They have 2 P alleles. Our heterozygous individuals are 2 times P times Q. There are 2 different ways that you can make this combination of 1 P and 1 Q. The P could have come from the mother, or the P could have come from the father. 2 different ways you can make that happen. Homozygous recessive, 2 copies of Q, the recessive allele. So P squared is equal to 0.7 times 0.7, which equals 0.49. So 49% of your population should be homozygous dominant. 2 times P times Q is 2 times 0.7 times 0.3, which is 0.42. Okay? So 42% of your population should be heterozygous. Now on the surface, you should not be able to tell the difference between these 2 individuals. If it's normal Mendelian inheritance, they should look the same. Okay, and this is why I gave you the illustration of eye color on Monday. I said it's not the greatest example because eye color is more than just 1 gene. It's actually at least 16 genes, but it tends to operate under this kind of a pattern mostly. Red-green color blindness is a much better example if you restrict it only to women, because men only have 1 copy of the X chromosome. It gets interesting. Okay, but homozygous recessive individuals are going to be 0.3 times 0.3, which equals 0.09. So 9% of your population should be homozygous recessive. Those are the only individuals that would look different. They would be colorblind in this case, colorblind females. Make sense? Okay, well the second one, what if the dominant allele increased to 90%? 90%, so now it's no longer 0.7, it's what? 0.9. Now it's 90% of all alleles. This one is no longer 1 minus 0.7, it is 1 minus 0.9, and now this is 0.1. 10% of all alleles are the recessive. So this one is no longer 0.7 times 0.7, it is now 0.9 times 0.9, which equals 0.81. 81% of your individuals are homozygous dominant. Okay, 2 times 0.9 times 0.1, 0.18. 18% of your population is heterozygous. But again, you would not be able to tell the difference between these 2. Okay, they would look the same. This is now 0.1 times 0.1, because the recessive allele only makes up 10% of the population. 0.1 times 0.1 happens to be 0.01, which means 1% of your population, 1% of those individuals should be homozygous recessive and would actually look different. So you're like, wow, I know what you're thinking. You're like, this makes up 10% of the total alleles. That's not bad. That's a pretty good percentage, and yet only 1% of the individuals would show that recessive allele. We'll come back to this concept next week more, and we'll talk about it a lot next semester of what that actually means. And it's called masking the recessive allele. So you have 18% of your population has at least one copy of that recessive allele. That's a pretty good amount of the population. And so you've masked most of it. So it gives you the illusion that it's not there, or at least not there in really high numbers. Let's say it's some kind of a genetic illness, and you're like, oh, cool, like a very small percentage of the population is born with this genetic illness. But then you have a huge percentage of the population that are carriers for it. Okay. Is that all right? Okay. Now number 3. I guess I could have left P and Q. Sorry, I got a little excited. Okay. So now for number 3, we have to do this a little bit differently. We have to work backwards. It tells us what Q squared is. Homozygous recessive individuals, right? These individuals are Q squared. Yes? They have 2 copies of the Q, the recessive allele. And it tells us that this is 0.25. 25% of the population is homozygous recessive. Right? Okay? So if we wanted to figure out what Q is, take the square root of both sides. Right? And we find that Q is equal to plus or minus 5. Plus or minus 0.5. But since we can't have a negative allele frequency, we will restrict that to not possible, and so we won't illustrate it. This is why it's wonderful to apply mathematics to real-life concepts, because then you get to ignore some of the weird imaginary stuff that you didn't get to do when you were taking algebra. Okay, so Q is equal to 0.5. We got that, right? Okay, so now if we wanted to figure out what P is equal to 1 minus Q, which equals 1 minus 0.5, and so P equals 0.5. They're both 0.5. Cool? Okay, so but that's not the answer. It wanted us to still give the genetic structuring. What are the genotype frequencies? Well, homozygous dominant individuals are P squared. They have 2 copies of the P, the dominant allele, which is equal to 0.5 times 0.5, which equals 0.25. It better, otherwise we did our square root wrong. And I don't ever do anything wrong. Oh, I love that you laughed. You're like, Dr. Ingo, you just told us 2 weeks ago we were getting a very, very bad version of yourself. Right? Do you remember when I told you that 2 weeks ago? It's just getting perpetually worse. The version of myself that you were getting is worse every single time we meet together. I'm sorry, I'm trying to stop it. All I can do is slow it down. It's just, I'm going towards somewhere, and that somewhere is scary. It's not good. All right. Heterozygous individuals, 2 times P, which is 0.5, times Q, which is also 0.5, is equal to 0.5. So 25% of our population is homozygous recessive, 25% of our population is homozygous dominant, 50% of our population is heterozygous. But again, you wouldn't be able to tell the difference between these two. So phenotypically, you would say 75% of the population bears the dominant allele. Okay? But as long as the population were at Hardy-Weinberg Equilibrium, you could do this genetic structuring just by counting the number of individuals that show that recessive trait. But only if the population is at Hardy-Weinberg Equilibrium. Because if it's not, then you can get some weird things going on because it's under some kind of an influence. And we'll talk about some of these things next week, but some of these we'll talk about, not until next semester. That's kind of a bummer. But again, you can't have too much because it's just too much good stuff is not good, right? Too much good stuff makes you sick and want to throw up, and I don't want that to happen when I'm around you. All right? Any questions about these problems? This is going to be something I'm going to expect that you're able to do. Yeah?
Okay, the only reason we were able to do that is because you chose sense you can't distinguish between.
Yes, so if I told you that it demonstrates the dominant phenotype, you'd be in a bit, you couldn't do it.
Because you can't distinguish between heterozygous and.
Right, you wouldn't know what to actually do. Then you would need to be given something in addition.
Okay.
You would have to, like, so if I told you 75% had the dominant phenotype, and the dominant phenotype makes up this percentage of the population, yeah, you couldn't do it if you were given dominant phenotype. You're right. And you also couldn't do it if the population weren't at Hardy-Weinberg equilibrium. The only reason you can do this to go from allele frequency to genetic structuring is if the population is at Hardy-Weinberg equilibrium. And so you have to assume that if you're trying to work out these problems. But, I don't know, just for fun, let's say I wanted to slip something in there that you should know, and you'd be like, oh, Dr. Engel, that's a trick question. Which usually means you didn't know your stuff as well as you ought to. If you're accused, anyways, we'll come to that later. I like to be accused of things that aren't true. If they're true, that's fine. You can accuse me of things that are true. But let's say I wrote you a problem and it said, you know, what is the genetic like for number 1? If the dominant allele makes up 70% of all alleles, what is the genetic structuring within the population if the population is not at Hardy-Weinberg equilibrium? Then you would just be like, can't solve it. Move on to the next one. Right? Because you would know that if it's not at Hardy-Weinberg equilibrium, you cannot get your genetic structuring from allele frequencies. Because weird things happen. Weird things happen when things aren't at Hardy-Weinberg equilibrium. Which this is a nice segue into the last framing question we're going to talk about today. When do populations not stay at Hardy-Weinberg equilibrium? Why wouldn't they be at Hardy-Weinberg equilibrium? And why would they be so cruel as to not allow us to do the genetic structuring? I mean, if y'all want to know, if y'all want some insight into why my brain is the way that it is, these are some of the things that I do all the time. Except for I deal with, like, populations where they have 12, 15, 20 different alleles for the same gene. But see, I don't do the calculation. I let a computer do it and then just do the analyses. But it messes me up. It messes with my brain. Makes me less equipped to actually talk to people in normal ways. So, I'm sorry. It happens. Somebody's got to do it, right? I'll protect you all from having this happen to your brain. All right. So population genetic structuring changes whenever there's evolutionary pressure. So whenever there's some pressure to change, to have some kind of microevolution where allele frequencies are changing, whenever there's some pressure for that to happen, the population can fluctuate. And so what's interesting is our assumptions of the Hardy-Weinberg equilibrium. You could say if any one of those is not met, it's going to be an evolutionary pressure to change allele frequencies. So our first assumption was that the population was infinitely large, right? Check this out. So genetic drift, these are random genetic changes. These are random genetic changes, and they're way more common when your population is small. Way more common when your population is small. If you have 10 million individuals in your population and you randomly lose 2,000, that's a small percentage of your population, right? Right? 2,000 compared to, what did I say before, 10 million? I don't even remember what number I gave. It just came to me. But let's say your population only has 250 individuals and you lose 50. That is a large percentage of your individuals, and you can have some wildly different gene pools as a result of depending on which individuals are lost in that random event. So some events that tend to shrink population sizes down and make drift a lot more powerful are events called bottleneck events. And the illustration here is like the idea of a bottle. So if you've got a big old bottle, like a Sparkletts water bottle, full of, I don't know, beads, right? Or full of marbles, or, I don't know, full of whatever you want to fill it with, as long as you can still pour them out, right? So it's full of something that you're capable of pouring out, and then all of a sudden you have some kind of event where you only pour some of the individuals out of that bottle. The rest of the bottle just disappears. So these happen, you see it happen in critically endangered animals when some kind of a natural disaster happens, and it was already critically endangered, and now you've dropped it even lower. And so you've shrunk your population down, you've lost potentially a lot of genetic variation, and at the same time now you have a really small population where random genetic changes are going to be a lot more common. What's cool is you can study the genetic variation within a species and figure out when or if and when there was some kind of a bottleneck event in the species history. And so we've done this with cheetahs. And cheetahs, you can go and you can see there was some kind of a massive bottleneck event in the history of cheetahs. And so if you took two cheetahs on the opposite ends of their ranges, they would likely be more closely related than you are to your siblings. Like there's that little genetic variation in cheetahs. So much so that virtually any cheetah could act as an organ donor for any other cheetah because there's so little genetic variation. You're like, wow, that's awesome, right? Like a cheetah never has to worry about having liver failure because you can just cut half of a liver out of another cheetah and give it to it until you realize that, well, one, cheetah livers, that's not the problem. The problem is cheetahs not having enough animals to chase down and kill, and we don't like when they chase down our cattle and kill them, and so, you've got yourself a big problem, a critically endangered animal. Yeah.
So how damaging is a bottleneck event?
It depends on how extreme the event is and how much of the variation you're able to maintain. So if you shrink your population down, let's say you lose 99% of your individuals in your population. And hopefully you don't have a population of 100 because that means you have one individual left. And if you can only reproduce sexually, you've got an issue, right? But let's assume maybe it's a good, healthy 10,000, 20,000 individuals and you lose 99% of it. As long as you maintain a good amount of the variation that was present there, it's not really that big of an issue. Drift is still going to be a more powerful factor in a small population than it was in a big, because there's a lot more, it's a lot easier to have some kind of random changes. But as long as you maintain that variation, the bottleneck event was not super catastrophic. If you lose a lot of your variation along with that drop in population size, now you have drift is going to be a lot more powerful of a factor in the future, and you're starting with very little variation. You've got two big problems. Yeah.
So basically, the bottleneck event happened, and the individuals that were left were all heterozygous. That would just keep the population going.
Yeah, that would be good. Yeah, if all you had are two alleles, and you lose. You lose a bunch of individuals, but all your individuals left are heterozygous, so you're still representing those two alleles. It would not be as bad as if you have a bottleneck event where all your individuals left are homozygous recessive. Yeah. Yeah. That could be very, very bad.
So I think it was cheetahs where they are, for some reason their population is going down. We can't understand exactly why. Could it be that
a lot of it has to do with little genetic variation?
Right.
So they just can't account for the way the ecosystem is changing. Ecosystems are dynamic, and they have very little genetic variation, and so they have a little ability to actually evolve in the future.
I know inbreeding within a family can be very harmful.
Yeah, and it's because of making certain recessive traits a lot more common, and a loss of genetic variation. But you can very quickly combat that issue. And so, there's a lot of concern when you take like an endangered species into captivity, like they did with the California condor. Like they took basically all living individuals into captivity to captive breed them. And so then there's like this issue of like, well, what do we do with the variability? Well, there are ways you can tackle that, and then there's a lot of things you can try to do to combat that and introduce new variation.
Eventually, could that lead to extinction?
Oh yeah, absolutely. Yep. Yeah, absolutely. I would say within 20 years, the cheetah will no longer exist in the wild,
You don't think
and
so?
it'll be only in zoos.
So how do you, when we talk about the flood, that was a major bottleneck.
Yep. Oh yeah. Oh, save that. Save that thought for 2 weeks from now when we talk about macroevolution. Yeah. But because microevolutionary changes have impacts in terms of macroevolution as well, and especially in terms of speciation. And that's what we're going to talk about next week. So save that question for next week and the following week. Yeah, Jake.
Could the bottleneck effect of a species lead into the chain reaction of another bottleneck effect of another species due to the food chain?
Yeah. Oh, yeah. Oh, yeah. You see that all the time with predators that are specialists that rely on a single prey item. If the prey population plummets, the predators plummets right after it, and you get this kind of predator-prey cycling that you see a lot, and in your specialized predators. Yeah, absolutely. So another time where this happens often is what's called the founder effect, when a small part of a population goes and starts a new or infiltrates a new area and founds a new population. Because oftentimes it's a small number of individuals, let's say like birds moving to a new island, or rats moving to a new island, or I don't know, fruit flies moving to a new island, whatever it is. And I keep using this idea of island because it's kind of hard to do this if it's not like on an island. You're not geographically isolated. It's possible, but you'd have to have some kind of other isolating mechanism. Oftentimes you have the individuals that go together are closely related. And so that you tend to, one, shrink your population down, and two, you tend to lose a lot of your variation. This is why you can get some weird things happening on island populations of species. And you're like, wow, this bird looks so strange on this island, but on the mainland it looks normal. And oftentimes it's due to genetic drift after the founder effect. All right. So the next assumption of the Hardy-Weinberg equilibrium was no individuals leaving or entering the population. So a way that you can have some evolutionary pressure is if you have gene flow, which is the movement of alleles into or out of your population, especially if you're bringing in new alleles. And this is one of the things that keeps island populations of some animals even possible, and to keep them from going to extinct because of really bad genetics, is occasionally you get new individuals coming in and introducing some new and novel variation. Yeah.
Would it be new individuals, or would it be just like metapopulations and stuff like that?
Metapopulations, different concept altogether.
Oh, it is?
Yeah,
Okay.
different concept altogether. So metapopulations are functionally a single population, and so
Okay.
there is some genetic structuring in
Yeah.
the different populations, but functionally
But
they're
it's still-
one. There's
okay,
so
'cause-
much gene flow that they're functionally one population.
Okay, got it.
And that's like humans, like with some exceptions of like some really isolated human groups.
Okay.
Humans are a metapopulation. And so it's like there's so much gene flow and movement of people around that it's basically one population.
Okay.
Now there are still genetic structuring in different
Yeah.
regions, so much so that you can use 23andMe and you can go and you can trace your heritage to specific regions, but it's not enough to do much.
So what if we had like a metapopulation and then like a geographic, something geographic happened which cut off that metapopulation? And so
Well, now you have something different. Yeah,
Okay.
now you have an isolated population, and genetic drift would be a really powerful factor, especially if you couldn't have any future gene flow. The third assumption was that there are no mutations, and so something that could provide evolutionary pressure is mutations are introducing new alleles, or they're adding more of a certain allele while eliminating another. And this was the example I gave you. If your mutation turns your dominant allele into your recessive, every time that mutation happens, it's converting one of the alleles making up your P into one of the alleles making up your Q, your proportion of dominant and recessive. Another one, our fourth assumption was that there's no sexual selection. So if sexual selection, so this is where one sex, usually the female of the species, chooses some phenotype in the male at the expense of others. And there are a number of reasons why you could do this. The textbook talks about one called the Handicap Hypothesis, where basically males illustrate something that is obviously a handicap. And so the example that you oftentimes use for this are peacock tails. And you're like, how is a peacock tail a handicap? But your experience of peacocks is probably in a zoo or wandering around a park somewhere that, they used to be part of a zoo, but they escaped because people didn't know that they could actually fly okay, and so they just kind of fly over the wall and disappear, right? That's your experience of peacocks. And you're like, well, why is this tail a handicap? But peacocks in the wild, that tail is a massive structure that's really easy for a predator to grab a hold of. And so the idea here is that the male is showing this female that there's this handicap that makes it really hard to escape predation, but I am so fit, so awesome, that I can still rock life in the midst of this handicap. And that's something that the female's like, wow, well, I want my sons to be able to have that kind of, vitality. Okay, and that's the idea of the handicap hypothesis. It's an interesting one. There are some other explanations for why this happened. Some of them are more interesting than others. There's one called the sexy son hypothesis, where it's just females are attracted to mates that they know will give them a higher probability of making sons that are also attractive to other females. And this is why in a lot of bird species where sexual selection is really powerful, the males are really pretty and the females are really dull. It's because the females are the ones providing the sexual selection and the males are the ones that are burdened by it. You have it a lot in fishes as well. All right, and then the last one, the last assumption of the Hardy-Weinberg is that there was no natural selection. And then so the other thing that can provide evolutionary pressure is natural selection. And this is the survival of the fittest, that some individuals survive better than others and therefore have a greater probability of producing more offspring because they survive better. That coupled with sexual selection, can give you not only do they survive better, but they also attract mates more. Although what you'll find is oftentimes sexual selection and natural selection act in opposite directions. Like with the peacocks. Natural selection favors males with shorter tails because they should be able to escape predators better. But sexual selection favors males with longer tails. What do you do? How do you figure out what's the pressure that's involved? It's hard. It's not easy. But it's worth it. All right. So here's an illustration of genetic drift. Here you've got your first generation. Most of your individuals have the dominant phenotype. If you look at this figure in the text, you'll notice that some of them are homozygous dominant, some of them are homozygous recessive, but they all show this dominant phenotype. And then what happens is only 5 of those individuals are able to reproduce for some reason. Maybe the other 5 died, or the other 5, just didn't find a mate or whatever. And then so your next generation, your proportion of homozygous recessive individuals dropped, and it had to do with just a random genetic change. And again, these are more powerful the smaller the population is. Smaller the population, the more powerful genetic drift is going to be. And then ultimately, if you've got these kind of random genetic changes, you have basically, there's only 2 fates for an allele, and one of the 2 has to happen. The allele disappears or the allele becomes fixed. Okay, so under genetic drift, you have 2 options. The allele disappears or the allele becomes fixed. And so what it means by fixed is every individual has only those alleles. And you see eventually all these individuals, capital B, homozygous dominant. You lost your recessive allele altogether. And they say red hair is disappearing due to genetic drift. That basically it's just random genetic changes and you have 2 options. It's either going to become fixed, which I would choose, right? I think everybody should have the opportunity to be redheaded, or it just disappears, and that's where it's actually heading, which is unfortunate. Yeah.
Oh, no, man.
Okay. Okay. Yeah. So here's the illustration of a bottleneck event. So inside of your body, you got all kinds of different forms, a ton of different genetic variation. But if you only pour a few of those out, the chances are you're going to lose a great deal of that variation. So not only did you shrink your population size down, but you also lost a lot of the variation. And lots of things cause bottleneck events: disease, natural disasters, competition with other species. We're really, really good at forcing animals into bottleneck events. You see this with like the American bison. You see it with California condor. You see it with the gray wolf, right? Things we either don't like them or we like them a lot, right? And so we kill a whole bunch of them, either because we don't like it or we like it a lot. California condors, we didn't like them because they're ugly. And I don't know, maybe we want to eat the dead animals that they're eating. I don't know. They are ugly. So here's gene flow. So you've got these 2 different populations. This population, they're all green. This population, they're all brown. This one's bored, right? It doesn't want to be in the all-brown population anymore, so it goes to the all-green, introducing new variation, something that wasn't there before, providing some evolutionary pressure for microevolution. All right, that's it. We're out of time. Enjoy the rest of your, what it is today, Wednesday?
Yeah.
Do you want to take your quiz?
No, I already took it.
Oh, yeah. What's up?
The discussion.
Yeah.
Cool stuff.
Oh, yeah. Population genetics.
Can I use this as my? Is there a specific one that's the correct answer? It's like the most.
No, not necessarily, as long as you can make a good argument for it, and that's what it really what I'm looking at. Yeah. Yeah. Yep.
Professor, so I have my study guide
All that fun.
for this exam.
Yeah.
Do you want me to meet with you to go through it? Or?
No, you don't have to.
I just like,
I do because I like voting.
It's up if you wanna. You wanna do your quiz, right? Yeah. So it's up here on the screen if you wanna just.
It's also fun to analyze them.
She just put the stuff in the slides in there.
Then you have
Yeah.
pretty much,
use
and then like,
graphs and whatnot
if
to
I
see
didn't
certain
understand something.
issues, stuff in the population, and graphs just.
Now you probably want to get a little bit more detail for some of these lecture break
I don't
questions.
want to learn about graphs anymore.
And so if you don't have more information on those, you know, you can
Just
grab
graphed it in, you graphed it out.
yeah, from other people that have.
Okay.
Yeah. Yeah. I tried to make this one simple.
Yeah, it was.
I don't feel bad though.
No cheating?
No cheating.
Oh,
You are very welcome. Oh yeah, sorry.
On Friday, do you have time to meet for classes for next semester?
I don't. Can you meet tomorrow?
What time?
Pretty much any time outside of 9:30 to 11.
Okay. Could we do like 1:30?
Yeah.
Okay.
At 1:30 I'll be in my office.
Okay.
So you can come find me there.
Thank you very much.
What time besides 1:30 now works best to me for my study guide?
What you got going right now? Okay.
Because I'm free after.
Come and find me. I'll be in room 20, which is just around the corner from here, at 3.
Okay.
Will that work?
That works.
Yeah, I'll be in there.
It's interesting. All of the animals that after the flood survived and flourished were very, very, I guess you could say, simple.
Yeah.
But the ones that were really highly derived, specialized, like the dinosaurs,
Right, which are going to have small amounts of variation.
variation survived, did not survive,
Right.
died out. So that's really interesting.
Yeah.
I've never made that connection. Makes sense.
Yep. No, so here's what happened. Yeah, the person was going to lead discussion asked yesterday if they could move to next week. So it wasn't enough time for me to get papers out, and then they obviously didn't get papers out because they were, so no. But it's okay because we've got a lot of nematode groups to talk about. You're welcome.